We'll get started with, okay, that's great. Okay, we'll get started with the event today. Really excited to have two great speakers lined up for microfluidics for fertility. So let's just get started with that. I'll share my screen. Okay, so first I would like to introduce BAM just a little bit. So we are a small team of just the five of us right now, and we've been organizing events to bring together the leaders in microfluidics from the Bay Area and beyond to foster innovation and collaboration, both in terms of these largest events and more intimate style events, which is our Friday Breakfast Club meetings. And so I would really like if we could just introduce the BAM organizers here on call to the audience. So I'll get started with myself. I'm Anushka. I'm currently a PhD student at UC Berkeley and really excited to be here with you all. Thomas, you want to go next?
Yeah, so hi everyone. I'm Thomas. I recently finished my PhD at Berkeley and I relocated to Colorado, and I'm now at Inscripta. Diego.
Hello everyone, my name is Diego Heike. I'm a fifth-year PhD student at Stanford. Sometimes you forget. I work in Juan Santiago's lab in microfluidics, and I've been in the BAM board for about a year at this point, like 3/4 of a year. And I think that actually we have, is Adam or Samira online right now?
They're not, no.
Okay, so I think that you can get the mic back, Anushka.
Yeah, great, thanks. But yeah, Adam and Samira are two more of our BAM members. Adam is an engineer at Inscripta, and Samira is currently a hardware engineer at Google X. Okay, cool. Diego, on to you for the mentorship.
Yeah, so we talked about this at our last event, but the Bay Area Microfluidics Network wanted to start a mentorship program because we noticed that there were a lot of people doing PhD programs that felt like they weren't communicating enough with people in industry. So the idea is that BAM helps leaders in industry. We have founders of companies, people who have been in industry for tens of years, and we connect them to PhD students, early stage PhD students, late stage PhD students. And we already started the program. We have around 12 members participating in the program, but we're looking to expand. And that is, we are looking for more mentors and we're looking for mentees. It's low commitment program. You just meet your mentor or you meet your mentee once a quarter. So it's about 4 times a year. And we're still looking for people who want to sign up. So in spring of next year, we'll connect more people. And so here's the interest form. It's just a Google form. It's a non-committal form. You can just tell us if you're interested in participating in the program, and we'll keep your information for spring of 2022. So the link is there, and I'll pass the mic back to Anushka, and then I'm gonna send the link on the chat for the Zoom meeting. Thank you.
Thanks, Diego. So one of the other programs that BAM organizes and that we want to talk about on the call today is the Friday Breakfast Club meeting that I referred to a few minutes ago. So we have these events that we have today, which is an event for a broader audience, a larger audience. And at FBC, we essentially ask a featured guest who is a leader of the microfluidics industry, has been working in the industry for tens of years. And we organize a call with our guest speaker and 9 participants, and those participants could be PhD students, could be people who have just started working or they're late in their career who just want to network and interact with the guest speaker. So we have an interest form here where you could either nominate yourself to be a guest speaker or nominate somebody else who you think would be a great guest speaker for the Friday Breakfast Club, and we would really appreciate it. Diego, if you could please post a link to this form as well on the chat, that would be great. Thank you. Right, and then finally, if you want to join us, there are a couple of ways, or more than a couple of ways, I guess. You can go to our website. You can contact us at hello@bayareamicrofluidicsnetwork.org, or we also have a LinkedIn group, which we would really appreciate if you could join that, since we post about all of our events there. So for tonight's program, we have 2 talks. one from Professor Utkan Demercy from Stanford, and the second one from Professor Bruce Gale from University of Utah. This would be followed by a discussion led by Dr. Dawn Arnold, who's also on call today. And then we will end this conversation with networking, where each speaker, Dawn and BAM members, will get their own breakout rooms. So all the participants could feel free to jump around the breakout rooms and join the conversations as they're happening. A few guidelines for the event today. Participants will remain muted until the networking session. And we really encourage all the participants to change their name to first and last name, followed by their affiliation, where they're working, if they're studying their university's name. If you have any questions, please type them in the chat and send them to everyone. These questions would then be asked during the Q&A session, as Dawn would ask them at the end of the discussion. And for the networking, we will be using breakout rooms. Hopefully, most of the participants are familiar with breakout rooms by now. If you have any questions, feel free to post them in the chat as well. And you would be able to change the rooms as per your own will across the 4 breakout rooms that we have. So yeah, thank you so much. Stay in touch. And now I would pass it on to Diego to introduce Professor Utkan Demercy for the talk.
Thank you, Anushka. So it is my pleasure to introduce Professor Utkan Demercy for our first featured talk for this event. And so Dr. Demercy is currently a professor with tenure at Stanford University School of Medicine and principal investigator of the Demercy Bioacoustic MEMS in Medicine Lab. So that acronym is BAM, which is the same as us. So love to hear that. And BAM is at the Cannery Center at Stanford for cancer early detection. Dr. Demercy received his BS degree in electrical engineering in 1999 as a James B. Engel Scholar, summa cum laude, from the University of Michigan, Ann Arbor. He received his MS degree in electrical engineering, MS degree in management science and engineering in 2005, and a PhD in electrical engineering in 2005, all from Stanford University. Dr. Demercy, you are co-host, so you should be able to share your screen and you can go ahead and get started. Sorry. Anushka, you can take it, Mike.
Okay, yeah, I think we will have the questions from the audience, but during the discussion section, later on after Bruce's talk, Dawn will then take over the questions, and you can have a more discussion-like answer to those questions, if that's okay.
Perfect, thank you.
Perfect, yeah. And Thomas, if you can now introduce Bruce.
Yeah, so thank you very much, Dr. Demercy. That was a great talk. And I definitely learned a lot, and it's very exciting. So next I'll introduce Dr. Gale. So Bruce Gale received his undergraduate degree in mechanical engineering from Brigham Young University in 1995 and his PhD in bioengineering from the University of Utah in 2000. He was an assistant professor of biomedical engineering at Louisiana Tech University before returning to the University of Utah in 2001, where he is now a chair and a professor of mechanical engineering. He is currently director of the Utah State Center of Excellence for Biomedical Microfluidics, a center devoted to research and commercialization activities around microfluidic devices. His primary interests include solving medical, biology, and chemistry problems using a variety of microfluidic approaches to complex and challenging medical and biological assays. Specifically, he is working to develop a microfluidic toolbox and approaches for the rapid design, simulation, and fabrication of devices with medical and biological applications. The ultimate goal is to develop platforms for personalized medicine, which should allow medical treatments to be customized to the needs of individual patients. As an outgrowth of his work, 6 companies have been formed, and he maintains a role at each. The first is Cartera, a multiplex instrument development company focused on protein characterization in the pharmaceutical industry that was spun out of his lab in 2005. The others include Aspira, which focuses on pathogen detection and exosome separations, Nanonc, which focuses on reproductive medicine applications of microfluidics, Wfluidics, which focuses on genotyping zebrafish embryos, and Microsurgical Innovations, which focuses on miniature medical devices, and Utah Nanoplate, which focuses on manufacturing of diagnostic microfluidic systems. So, Dr. Gale, looking forward to hearing your talk. Thank you very much for joining.
So you're muted right now.
Got so excited to start. Thanks for pointing that out. Yeah, I appreciate the introduction, and I appreciate Dr. Demercy's talk that covered a number of things that I was going to introduce, so that'll save me a little bit of time as well. I'm going to talk to you a little bit about some work that we've done related to using spiral channels to do sperm preparation. And I should maybe say up front, we didn't invent spiral channels or anything like that, but we've figured out some ways to use these channels in unique ways for these fertility sorts of treatments. And this work started 6 years ago now, when the andrology department approached me about some potential work they were hoping to do, and so this kind of grew out of that. So I want to make sure that I've worked with them, and I'll recognize them a little bit later. But we use these spiral channels to do these separations, and if you're familiar with inertial microfluidics, you may recognize these devices. I'll talk just a little bit about how these ones in particular work a little bit later, but the general concept is that you can use them to basically focus beads, sperm, other things like that inside these channels. And so that's kind of the general concept. And before I move on, I just want to make sure you're aware that I do have financial interests in the technologies that are presented here. So I'm going to, art's a big deal. So assisted reproductive technology, Dr. DeMercy covered that. So I'm going to skip that. He also mentioned why, which is we're going to address two fertility sorts of treatments here. One is IUI, and then the other one's processing of micro-TESE, and I'll explain what that is in a minute. But IUI is kind of like the first sort of treatment that you would do. It's the simplest, and it basically just cleaning up the sperm sample, if you will, and then re-injecting that sperm sample into the uterus to hopefully generate a child. And there's the overall process is pretty simple. And we collect semen, we process that semen, and then that's injected. And so I'm just going to focus on some of the processing of the semen. And so that'll be one of the projects I discuss. And then the second one is going to be this micro-TESE. So this stands for a micro dissection testicular sperm extraction. This is essentially a biopsy, or to make it simple, they stick a needle into the testicles, into the areas where they think there might be some sperm present, and they try to pull out the sperm from these areas of the testicles. Now, this is a pretty extreme surgery, if you will, and it's only done if there's essentially no sperm that are being found in the ejaculate, in the semen, things like that. So this is the only, I would call it a kind of a last ditch effort to see if they can find a few sperm that they might be able to use to do ICSI. And so this is, again, or if they can find enough, they'll do in vitro fertilization, but most of the time it's an ICSI sort of thing where they have one sperm. And when you do a micro-TESE, collect a micro-TESE sample, you might imagine that there's going to be all kinds of blood cells in there. There's other sperm precursor cells, epithelial cells, all kinds of, I'm just going to call it junk, things that we're not interested in. So in that case, you have sperm that are in very low concentrations, usually 20 to 50 per 10 to the 9th cells. So this is that proverbial needle in a haystack where you really can't, it's difficult to find these individual sperm cells. And then the other thing that happens is they tend to be relatively immature, meaning they haven't gone through the process that allows them to swim. So you're not going to find them because they're swimming. So a lot of the existing technologies that depend on sperm swimming to identify them just don't work, because these are sperm that are not, if you will, fully developed in the sense that they can swim, but they still can be used for ICSI and some of these other in vitro fertilization sort of applications. And so we worked with the andrology department to find these sperm essentially. So how does it work? They basically collect the sample, they do tissue dissociation, basically take a razor blade and kind of cut it up, and then they manually search for sperm under a microscope. As you can imagine, this is pretty difficult. So this is a technician just basically staring through a microscope for long periods of time, 8, 10, 12 hours sometimes, to see if they can find sperm in these samples. And if you were to look at a normal sample, you'd see that, you know, these sperm are pretty easy to find, but in situations where there's not really many sperm present, I mean, this is, you know, the Where's Waldo sort of thing, right? Where's a sperm in this mix that's just full of blood cells and other things like that. So you have challenges with error, and, you know, really have to be a pretty skillful individual. So that makes it expensive. And so this is not really an ideal scenario. So that's kind of where we were coming from. How can we reduce this effort required to do this search? So we decided we might try the spiral channels to make this work. These, if you imagine, there's also some situations where you have sperm and white blood cells that are just mixed together. That's called leukodystrophy. But in this case, we have white blood cells, red blood cells, and a bunch of other things. And this is kind of the concept that you'd put them in one end of the spiral channel, and at the end, you know, all the red blood cells, white blood cells, junk cells go out one side, and the sperm would come out the other. So you can basically clean up the sample. So that's the concept, and we'll talk about how it works. So the general principle is that you have, you know, your laminar flow distribution, you have a shear stress distribution, and the idea for particles in this situation is the shear lift force is going to push it towards the wall, and then there's a wall repulsive force that, or a lift force that pushes it towards the center, and they tend to move towards some equilibrium position. So this is kind of standard inertial microfluidics. If you do this in a circular tube, you're going to get kind of a ring of particles. In a square tube, you get these four, and in a rectangular tube, you get these two focusing positions. And again, these are for spheres. So if you just have spherical particles, now you add in the Dean flow. This is the, I guess, the countercurrent flow that sets itself up in a spiral. And you can imagine now by looking at this, if you have a rectangular tube that has, you know, two focal areas of sperm or spheres in this case, the Dean flow is going to cause them to move towards one wall or the other, depending on where they are. So if they're in the center of the channel, they move towards the outside. If they're closer to the, you know, the top or the bottom of the channel, they'll move to the inside. In this case, the spheres tend to focus a little more towards the top and the bottom, so they move to the inside. So that's kind of standard inertial microfluidics, and that's for spherical particles. Sperm are not spherical, so this is it created some interesting problems as we jumped into this work. Typical sperm has about a 5 micron head with about a 50 micron tail, and as Dr. Demirci pointed out, they can have actually a wide variety of other problems, sizes, shapes, all kinds of things. Biology is not consistent by any means in many cases. So we've got these highly asymmetric particles. What happens to them? Well, first thing that it does is the sperm tend to line up just because of their shape. So that creates some interesting effects. But they do tend to focus as well. In this case, interestingly enough, they all focus to the center, which we'll talk about why that is in just a minute. But because of their shape and their alignment and other things like that, the process pushes them into the middle. And that can be used to, and we just found this by experimenting. It wasn't something that was necessarily predicted. So now when you put them in the Dean flow, instead of them going towards the, you know, towards the inside, they're going to get pushed towards the outside because they're in the middle of this channel. And that allows us to do separation. So all of the kind of more spherical things end up towards the inside of the channel. The sperm tend to move towards the outside of the channel, and we use that to do our separations. So we can, you know, select one side and, if you will, not select the other. These are fairly standard channels in PDMS, so I'm not going to spend too much time on that. Some of the micro-TESE experiments I'm going to show you here were with 50 tall, 200 micron wide channels, and with 3 loops. The IUI channels we did were actually quite a bit larger, and I'll talk about that in a minute. Just so you kind of know how we do the analysis, this is an example of 10 micron beads in a channel, and we just measure the fluorescence across the width of the channel, and that gives us kind of an idea of the focusing position and then how tight the focusing is. And these are some examples of sperm and red blood cells in this case, showing that we can move them to different locations. And as we implement this, you can see that if we take the selected portion, we get a lot of sperm in the selected portion, and the unselected portion maybe has 2 sperm. So in this case, it's not a perfect separation, and there's a variety of reasons why that might be the case. But we do see that we get a high concentration of sperm in this selected portion. So it actually seems to be working, and we can pick one part of the sample that has sperm in it. Without going into too much detail, there's things with the Reynolds number. As you increase the Reynolds number, you can change where sperm focus a little bit and how tight the focusing is. You can, anyways, focus red blood cells in different locations than, say, white blood cells and other features. But this is just one example here of, if you look at the channel, we have sperm, blood, or sperm tend to be towards the inside. Blood cells tend to be the outside. We actually did another test with cryptosporidium just for a totally different project, but that's in here as well. They show they go to the outside as well. Anyways, we can kind of push the sperm towards one side and get most of them to the inside. And there are a few that, for whatever reason, focus to the outside, and they may, sometimes they don't have a tail, or they're, the tail's wrapped around their head, or a variety of other things. Make a long story short, in the interest of time today, we have some examples of actually implementing this the system. So this is a micro-TESE sample that was run. If you just take it, and in this case, we took a micro-TESE sample and we injected some sperm into it. So we added a few sperm in there just to, verify, make sure we knew that there were going to be sperm. And you can see that the unprocessed sample have 1 or 2 sperm in it, and the processed sample in this case has a much higher number, usually about 10-fold is what we found. Oh, sorry. And we took a number of other samples from the clinical samples that we had, and these were kind of fun to run. We were able to show that with 8 different samples, you had some that had maybe you found 1 initially, but we could find 8 after we processed it. 23, 136. These ones are much larger numbers, but in every case, we were able to increase the ability to find the sperm dramatically by factors of, like I said, usually about 10, 5 to 10 in most cases. So that was, if nothing else, that technician that's sitting there staring at the sample will have a much easier time finding these sperm cells, and so that's one of the key things. And then there were at least a couple of examples, I don't show them right here, where we were able to find a sperm cell when they weren't unable to find sperm at all in the unprocessed sample. And that's the goal here. Suddenly there might be able to have a pregnancy because you found a sperm cell out of an individual that had no sperm that were otherwise identifiable. So that's a real opportunity, and kind of where we're trying to go with this. So just quickly, we took this and thought, well, hey, we can just process normal sperm samples as well. If we can do micro-TESE tends to be in, I'm going to call it more normal fluid samples, but semen samples, as you might imagine, are not entirely normal. They tend to be very viscoelastic. And we found that you put sperm into a viscoelastic channel and they don't behave the same way. All the sperm, in fact, the viscoelastic forces tend to push all of the sperm and every other thing into the center of the channel in this case. So I'm going to maybe run through this a little bit quick, but so what we ended up doing, so anyways, we suddenly couldn't separate things when we used normal sperm samples, because semen samples, because just everything got pushed into the middle. This process that worked for IUI suddenly didn't work for a semen sample. So what we ended up doing was we ended up running these through the spiral a couple times to achieve a couple of things. One, we were able to put in, a liquefied semen sample and some media, process that. And as you do that, it goes from 50% of the, the semen environment down to 25%. And you run that a couple times and you can get it down to less than 1.5%. And so the viscoelasticity drops off dramatically as you go through that process. And so if we do that, once we get it below a certain value of viscoelasticity, then our separations go back to functioning normally, and we can separate sperm one way and everything else the other direction. So this was, in this process, we were able to remove all of the junk cells and just essentially keep most of the desired cells, and then also reduce the sample volume. So when you do an IUI, you only want about 1 milliliter of sample. So we start with, by liquefying the sample, you can separate out the sperm cells from other debris, especially white blood cells and red blood cells. We can also wash them from all the prostaglandin containing seminal plasma, which you don't want because the female body doesn't respond well to those. And then we can reduce the sample volume by, I guess I should have I got ahead of myself there, by running some additional cycles. So we run this a few more times and we can eventually reduce the sample. So we, in this case, just by running it through this same spiral channel about 7 times, we can take a sample, collect about 80% of the sperm in this case, reduce the prostaglandin containing seminal plasma, and give you a volume that's the size that you need for IUI. And so that would essentially, and you're just pushing this back and forth through the spiral channel. So it's fairly simple. So we built an automated instrument to do that. Here's kind of an early prototype of that. And when you run the protocol, we get about a 70% modal sperm recovery. If you run it, you can rerun the waste and actually push that up to about 80% typically. And then the motility is good compared to density gradient, and the viability is good. The DNA damage is similar. So the point here is that the automated instrument doesn't do anything that normal processing already doesn't do. So it should be something that you could use. So the summary is we've got these devices that we can use to process micro-TESE samples. We've adapted that same process for IUI samples, and then we have an automated instrument that can run these. So I'd like to acknowledge, the funding sources for this work. These are the andrology collaborators here at the University of Utah. And then of course, the students that do this work were mostly, or anyways, they do most of the work. I just watch them. I should point out Jiyoung Chang, or Jiyoung Sun. We have a faculty member named Jiyoung Chang, Jiyoung Sun, who's I think on this call, so he can answer all those questions that relate to his work. And then Alex Jafik and Rahil Samuel, they were the primary folks that did the work here. So anyways, happy to answer any questions. So.
Yeah, thank you very much for that. That was fantastic. The mountains are beautiful there too.
That's right. That's what it'll look like next week after it snows, but it's not quite this week.
It's gonna be about the same over in Boulder. Yeah, so next up, we'll open our discussion section. And Samira, are you ready to introduce Dawn?
Yes, thanks to both of our speakers. This was great, and I can't wait for the Q&A. And without further ado, I'm going to introduce you all to Dr. Don Arnold. Don received his BA in chemistry from Cornell University and his PhD from UC Berkeley, studying chemical reactions and molecular spectroscopy. From 1997 to 2000, Don worked at Sandia National Labs, and he focused on microfluidics and microchemical analysis of systems. In 2000, Don co-founded a spin-out company based on this technology, Exigent, which was acquired by AB Science in February of 2010. In 2012, Don became VP and R&D and principal scientist in AB Science, and Don transitioned to business development in 2015, identifying technologies showing potential for SciEx pipeline. In 2017, Don founded his own company, Veristat, where he provides technical and business consulting for life sciences companies that navigate from startup to exit. So if you have any questions about taking your startup to exit, go to Don, and Don, please go ahead and start the Q&A.